Alright, so we're back with our third section on cells and the structures within cells, taking a look at vacuoles, which is a structure that typically animals do not have, but plants, protists have it. Typically, kind of those are the two main groups that would have those. There can be different kinds of vacuoles. The biggest, most famous one is the central vacuole, which you find in most plant cells. This is the main part of a plant cell on the inside, in addition to the cytoskeleton that helps maintain structure. And as plant cells grow, typically one of the ways they grow is by adding water to that central vacuole, causing it to fill up, and as a result, the cell will actually get bigger. So it's involved in growth, it's involved in structure. Other kinds of plant cells can grow or maintain other things inside their vacuoles as well. They can store toxins, they can store pigments of things. Protists sometimes will use vacuoles for movement. So what they'll do is they'll actually take their vacuole, fill it up with water, and then push that water out really, really hard, and that will cause the cell to move forward through its environment. So that's called a contractile vacuole that you find in some of the protists. Storage vacuoles, again, I mentioned some of the ones before. Growth vacuoles, storage of all different kinds of things like toxins or poisons, even chemicals that are produced by a cell that might become toxic if they built up. They can be stored safely inside of a vacuole so that it doesn't hurt the rest of the cell. And even things like flowers that have pigments, they can be used to attract various other kinds of pollinators, for example. Those pigments are oftentimes stored up in vacuoles. Now, microbodies, there isn't just one category of microbody. There are lots and lots of different kinds of microbodies. One of them is called the peroxisome. And peroxisome, again, one type of microbody is one that contains a substance called catalase, which is an enzyme that helps break down something called hydrogen peroxide. So H2O2, hydrogen peroxide, is toxic to cells. It is something that cells often will produce as kind of a byproduct in some conditions. And unless they have some way to break it down, there's a chance where it could become toxic and actually cause damage or even kill a cell. So catalase is an enzyme inside these microbodies that's used to break down H2O2 before it builds up to the point where it might become toxic. Mitochondria are used in energy processing. They, again, are found in all different kinds of eukaryotic cells, not just animals. They're in animals, plants, fungi, and protists. They're involved in breaking down things like glucose to produce molecules like ATP for energy. Fairly complicated little structures. There's an outer mitochondrial membrane. There is a space called the intermembrane space. And anytime you see the prefix inter, it means between two or among three or four different things. Intermembrane space is a space between the outer mitochondrial membrane and the inner mitochondrial membrane. All these little wrinkles and folds on the inside of this mitochondria, these guys here, all these little folds here are called cristae, and what they're going to do is they're going to help produce more surface area this mitochondria can use when it comes time for it to do its metabolism. And then all the way in the center of this thing, all the way on the inside of the inner mitochondrial membrane, like right in here, is a space called the matrix. And that's where some of the really important parts of oxidative metabolism are going to take place. So oxidative metabolism means using oxygen in part to break down glucose and produce ATP, which is the primary energy currency for most cells. Mitochondria are one of the main ones that do this, especially when it comes to oxidative metabolism. Mitochondria are the really, really important players. So again, just a quick look at mitochondria. Mitochondria also do have their own little bits of DNA. These little circles here are DNA. They also have their own ribosomes inside of here, which is kind of unique. Not a whole lot of organelles have their own ribosomes or DNA. And kind of the interesting part about this is these ribosomes and little bits of DNA are actually more bacterial than they are eukaryotic, which kind of gives us a clue as to how they might have came to be many, many millions and actually billions of years ago. All right, chloroplasts are the other main organelle involved in energy processing in cells. And again, these are involved in plants. They can be involved in protists, but that's really about it. Animals and fungi don't have chloroplasts. It's only plants and a few of the protists. And so primarily the main pigment we're dealing with here, the main substance inside the chloroplast that we deal with is one called chlorophyll. And there are multiple different kinds of chlorophyll, but we're just going to keep it kind of generic for now. Chlorophyll is a pigment which is able to capture photons of light and use them. Use those photons of light to kick off a chain reaction of events that eventually lead to the production of glucose. Now, chloroplasts, kind of like mitochondria, also have a pair of membranes on the outside. There's an outer and inner chloroplast membrane, just like we saw with mitochondria. But chloroplasts are different beyond that. Chloroplasts have little discs inside themselves called thylakoids. Think about a thylakoid as being like a little green Mentos. Mento? Is one Mentos a Mento, or is it still Mentos? I'm not sure. It's a little green mint-shaped structure that's involved with containing chlorophyll, capturing sunlight, and helping to eventually process that into glucose. If you have a stack of thylakoids, that would be then called a granum. If you have multiple stacks, that would be called grana. The thylakoids are the really important part involved with what's called the light-dependent reactions of photosynthesis. They also have their own DNA, just like mitochondria do, and that DNA does look a bit more bacterial than it does eukaryotic. The other substance we need to take a look at with a chloroplast is outside the thylakoid discs, in this space out here, and that's called the stroma. The stroma is the space outside the thylakoids. That's where the second half of photosynthesis takes place. That's where what we call the dark reactions takes place, is in the stroma. So again, two membranes, stacks of thylakoids called grana, ribosomes, DNA, and stroma outside those discs. All right, now onto the cytoskeleton. Cytoskeleton again can be found in any and all of the members of the eukaryotic kingdoms. They're made out of proteins. There are different sizes. There can be what are called microfilaments, intermediate fibers, and microtubules. And from left to right, that's what we're seeing here in this picture. Microfilaments, intermediate fibers, and microtubules. So a lot of functions here. A cytoskeleton is involved in cell shape. If you look at an animal cell, that's the only rigid structural support an animal cell will ever have is from the cytoskeleton, because they don't have a cell wall or a vacuole. Plants have those things. They have a cytoskeleton too, but they have a lot of other things to help support their structure. Animals don't. Cytoskeleton components can also be involved in anchoring organelles in place, preventing them from moving around too much, almost like a tie-down strap or a ratchet strap you could use to keep cargo in place in the back of a truck or a trailer. Cytoskeleton can be involved in movement. It can be involved with flagella and cilia, and also in cell division, mitosis and meiosis. The cytoskeleton is involved there as well. So it's a very dynamic system. These are proteins that are made up of amino acids, but they can be broken down and rebuilt. The cytoskeleton, very, very loosely, is kind of like the frame of a house. It keeps it upright, keeps the shape intact, hopefully, if things are going the way they're supposed to go. But beyond that, the cytoskeleton is much more dynamic. Like the 2x4s and dimensional lumber in your house is pretty much the way it was whenever your house was built. Cytoskeleton, on the other hand, can change second by second. Individual microfilaments, individual microtubules can be taken apart and rebuilt just as quickly to form new structures inside of a cell. So again, it's a very dynamic and changeable kind of a system. Microfilaments, again, are these smaller solid rods made up of proteins like actin, twisted together. They're involved in cell shape, they're involved in cell movement, especially when it comes to cell crawling, which is something some organisms like amoebas can crawl from place to place using their microfilaments. And also cell division. When it comes time for animal cells to divide, they're going to use microfilaments to form what's called a cleavage furrow and divide that one cell into two cells. There are also intermediate fibers, which are medium-sized, oddly enough. They are hollow tubes, also made out of proteins. They also help to support the shape of the cell, as do all the parts of the cytoskeleton. But they're also involved in keeping organelles anchored in place, like those ratchet straps I mentioned a second ago. These are the most permanent parts of the cytoskeleton. They typically do not get broken down and rebuilt like the microtubules and microfilaments. Microtubules are the biggest in diameter. They're hollow. They are again involved in shape, but also cilia, flagella. And one function I didn't mention a few minutes ago on that previous slide was that part of the cytoskeleton is involved in helping organelles move. So like we saw transport vesicles, moving from point A to point B inside of a cell. They don't just do that randomly. They're not floating around inside the cell like a bubble. Well, that's kind of fun to think about it that way. They're actually attached to parts of the cytoskeleton, almost like a monorail, so they can go back and forth inside the cell to where they need to go. So microfilaments, again, involved in cell crawling. This is an amoeba. These would be bacteria. This thing is very, very gradually creeping up and will surround these bacteria and eventually digest them. This is microtubules showing you the movement of a vesicle back and forth along the length of this hollow rod. So it's a pathway, like a monorail, for these vesicles to go from point A to point B. We can also look at movement. Cilia versus flagella. Cilia tend to be present in relatively large numbers. Cells that have cilia tend to have hundreds or thousands of them. And because they have so many, it becomes really critical that they cooperate with each other. If you think about a cell covered in hairs, little tiny fine hairs, if those cells don't all cooperate with each other, if they all just start beating whenever they feel like it, that cell is going to swim around in a circle, it's going to look kind of funny, and all the other cells are going to make fun of it. You think about like the old like movies or cartoons, and you've got like a big Viking warship, and you've got the guys, with the oars, and they're all kind of moving the oars at the same time. You got that big dude up front pounding on the drum, he tells them when to row. Why are they doing that? Right? Do they just like the beat or like the rhythm? What is it? Well, the rowing needs to be in sync. Otherwise, that boat's going to go around in a circle and it's going to get blown up or boarded by somebody, or something bad's going to happen to it if it's not going in a straight line. Just like in a cell with cilia, those cilia have to link up and coordinate if that cell is going to go from point A to point B and not just swim around randomly. That movement of cilia can cause a cell to move. We see things like paramecium, which is a protist moving using cilia. But on the other hand, we can also think about cells that move things across their surface using cilia. So, for example, in your upper respiratory tract, the cells in your upper respiratory tract, some of them have very, very fine cilia that are able to move things out of your upper respiratory tract. Those cells are often surrounded by other cells that produce mucus, which serves as kind of like a filter to keep stuff out of your lungs that maybe you don't want down deep inside your lungs. Things like dust or pollen or your neighbor's skin cells. You really don't want those down inside your lungs. So what happens here is the mucus traps those particles, and the cilia moves the mucus and trapped particles up and out of the upper respiratory tract. If someone is a very, very heavy smoker, after a certain period of time, those cilia become paralyzed. They can't move anymore. Right? You're breathing in smoke, which is basically made up of little tiny bits of dust, which get trapped in the mucus. The cells continue to make more mucus, but the mucus can't go anywhere because the ciliated cells are all paralyzed. So what does that do? That creates that very nice, thick, heavy smoker's cough that everyone finds so attractive, right? That happens because the ciliated cells have become paralyzed due to the cigarette smoke, and therefore they can't move the things out of the upper respiratory tract that they should. Now, flagella, on the other hand, typically only a cell will only have one or two of these. If a cell has any flagella at all, it's going to have one or two, typically for movement. And a classic example of this one would be like a sperm cell, which has a single flagellum, at least in animals, to allow it to go from point A to point B. So again, ciliated cell, this would be a paramecium, not normally lime green, but this one has been stained so you can see it more clearly in the picture. Lots and lots of cilia covering the outside of that cell. And again, a sperm cell with a single flagellum, right, causing it to move to where it needs to go. All right, so at that point, we are just about done with the organelles. We have one more little section on attachment, but I've got a couple of slides here that might be helpful to let you review. And so the idea with these slides, again, is if I sort of go back here a second, we take the manufacturing functions. Right? If you quiz yourself, if you say, okay, self, there are about 4 different organelles that make something inside the cell. What are they, and what do they make? And there's actually more than 4, but that would be a good practice short essay type question. Can you think of 4 organelles inside of a cell that build something? Tell me what they are, and then tell me what they build. Right? So you've got things like the nucleus. What does it build? Well, it makes mRNA. It's involved in making ribosomes. It's also involved in making new DNA from time to time. You've got the rough ER. What does that make? Well, the rough ER can make proteins. The rough ER also manufactures membranes. What about the Golgi? Well, Golgi is involved in protein production. What about smooth ER? Well, smooth ER is involved in making lipids. You could also say ribosomes, involved in making proteins. What about organelles that are involved in breaking stuff down? Well, you could obviously, you could go with lysosomes. That would work. You could go with peroxisomes. That would work as well. You could even go with vacuoles, because sometimes vacuoles have the ability to break down toxins too. Okay, what about organelles involved in processing energy? Well, the 2 that come to mind for me are the chloroplast and mitochondria. They seem to be the 2 that we talked about that are involved most with converting energy from one form to another. So we've got those 2. And then just general other functionality, we've got things like the cytoskeleton, we've got things like the cell wall, we've got things like central vacuoles. Those could also play a role as well for just kind of general functionality. This is a list of all the organelles we talked about, I think, except for peroxisomes, maybe. Yeah, so the different functions we talked about for all of them to kind of help you review. It's like a big review slide. 3 jobs of the smooth ER. What are they? What's the 1 job of the mitochondria? What are 2 functions of the plasma membrane? We talked about 4 or 5 of those. So if you can do this, you should be in pretty good shape when it comes to the organelles. All right, last section, cell-to-cell communication. There are three types of cell-to-cell connection and communication. The first type is called an anchoring junction. Anchoring junctions tend to be in parts of the body where you need to have a lot of physical strength. These cells are like riveted together. You've got a protein on the inside, you've got a protein on the outside, protein on the outside, protein on the inside. And these proteins are all linked together. It's going to be really hard to separate those two cells from each other. So in skeletal muscle, where there's a lot of stress and strain, in skin, where there's a lot of stress and strain, it's very much to your benefit to have anchoring junctions in those parts of your body because they're subject to the most mechanical physical stress. Tight junctions are where you want to have leak-proof cells. So now we don't really have the proteins linking the cells together like rivets. We've got the proteins pressing the cells together, right, very, very tightly. So what that's going to do is it's going to help prevent leakage of materials from whatever tissue this is into other tissues. Right? So we've got tight junctions helping to maintain waterproofness of tissue. And then finally, gap junctions are where we have openings between cell membranes, and in some cases for plants, cell walls, to allow communication. If it's a gap junction in an animal, it could be a place that we see like in cardiac muscle, heart tissue. Those cells have gap junctions to allow electrical signals to pass from one to the next very, very quickly, with very little time delay involved, so that the heart can actually beat as a single functional unit. We can see in plants, we can see other structures called plasmodesmata, openings between cell membranes and cell walls to allow movement of nutrients, sugar, minerals, those kinds of things, hormones from cell to cell, because they're not completely walled off from each other. There are these gaps to allow molecules to travel back and forth and allow the cells to not only communicate, but share materials that they might have encountered. All right, so that's the end of section 3. I will get this up, posted as quickly as I can, and we will move from there into chapter 4.